Development. Skeletal, cardiac, and smooth muscles are derived from the mesoderm. More specifically, skeletal muscle is derived from the paraxial mesoderm. Cardiac muscle is derived from the lateral splanchnic mesoderm. Smooth muscle fibers differentiate from the splanchnic mesoderm. Smooth muscle exceptions not derived from the mesoderm include the muscles of the iris, sphincter and dilator pupillae, and the myoepithelial cells in mammary and sweat glands, which are derived from mesenchymal cell originating from ectoderm. Organ systems involved. Organ systems with striated muscle. 1. Cardiac system, heart. 2. Skeletal system, muscle attached to bone. 3. Gastrointestinal system, upper esophagus, external anal sphincter. 4. Urinary system, external urethral sphincter. 5. Visual system, extraocular muscles. Organ systems with smooth muscle. 1. Integumentary system, arterpili. 2. Gastrointestinal system, walls of hollow visceral organs, for example, intestines, pancreas, liver, gallbladder. 3. Respiratory system, bronchi and bronchioles. 4. Urinary system, ureters, bladder, internal urethral sphincter. 5. Reproductive system, uterus. 6. Visual system, ciliary muscle. 7. Vascular system, blood vessels. 8. Lymphatic system, lymphatic vessels. 4. Urinary system, ureters, bladder, internal urethral sphincter. 5. Reproductive system, uterus. 6. Visual system, ciliary muscle. 7. Vascular system, blood vessels. 8. Lymphatic system, lymphatic vessels. Function. The primary function of skeletal muscle contraction is to allow for the performance of specific movements. Skeletal muscle also provides structural support, maintains the body's posture, stores amino acids, and maintains core body temperature via shivering. There are two types of cardiac muscle cells, autorhythmic and contractile. Autorhythmic cardiac cells do not contract, they set the pace of contraction for other cardiac muscle cells, which the ANS can modulate. Contractile cardiac cells, cardiomyocytes, constitute the majority of the heart muscle and can contract. There are two types of smooth muscle cells, single unit and multi-unit. Single unit smooth muscle cells are found in the gut and blood vessels. They are linked together via gap junctions, allowing for contraction as a functional syncytium. They also contract myogenically and can be modulated by the ANS. Multi-unit smooth muscle cells are found in the muscles of the eye and at the base of the hair follicles. They contract by being separately stimulated by nerves of the ANS and allow for fine control and gradual response. Mechanism. Types of striated muscle contraction. There are 4 types of striated muscle contractions: isometric, isotonic, concentric, and eccentric. Isometric striated muscle contraction is characterized by a change in muscle tension without change in muscle length. Isometric contractions are seen when pushing against an immovable object or trying to lift a weight that is too heavy. Isotonic striated muscle contraction is characterized by a constant muscle tension without a change in muscle length. This type of contraction occurs when the contraction force matches the total load on a muscle. Isotonic contractions are seen during activities such as walking, running, or squatting. Concentric striated muscle contraction occurs when there is sufficient muscle tension to overcome the load, and the muscle contracts and shortens. During this type of contraction, a muscle is stimulated to contract according to the sliding filament theory. Concentric contractions are seen during activities such as a biceps curl or standing from a squatting position. Eccentric striated muscle contraction occurs when the muscle works to decelerate a joint at the end of a movement as opposed to pulling it joining in the direction of the contraction. This type of contraction can occur involuntarily, for example, while attempting to move a weight too heavy for the muscle to lift, or voluntarily. For example, when the muscle is smoothing out a movement or resisting gravity, such as during downhill walking. Eccentric contractions act as a braking force in opposition to a concentric contraction to protect joints from damage. General mechanism of skeletal muscle contraction. Skeletal muscle contraction initiation and execution occur in the following steps. An action potential, AP, travels along a motor nerve to its endings on muscle fibers. At each motor nerve ending, the nerve secretes acetylcholine, AC. AC acts locally on the muscle fiber membrane to open AC-gated cation channels. The opening of AC-gated channels allows large quantities of sodium, Na, ions to diffuse to the interior of the muscle fiber membrane. This action causes a local depolarization, leading to the opening of voltage-gated sodium, Na, channels, which initiates an AP at the membrane. The AP depolarizes the muscle membrane, causing the sarcoplasmic reticulum, SR, to release large quantities of Ca ions stored within the reticulum. The Ca ions produce attractive forces to act between actin and myosin filaments, causing them to slide alongside each other, leading to the contractile process. After a fraction of a second, the Ca ions are pumped back into the SR by a Ca membrane pump and remain stored in the SR until a new muscle AP occurs. The removal of Ca ions from the myofibrils causes muscle contraction to cease. Intracellular Ca is then removed by the SR, dropping the concentration of intracellular Ca.